Welcome to our lesson on the human digestive system. The key questions for today are 1. What organs make up the digestive system? 2. What is the path of food through the digestive system? 3. What are enzymes? 4. How do enzymes help digestion? 5. What factors can affect enzyme activity? The key words you will need to know are digestion, soluble, lipase, protease, carbohydrates. The digestive system is an important system in the human body. The purpose and function of the digestive system is to break down the food that we eat into small, soluble molecules that can be absorbed into the bloodstream. These molecules can then be transported around the body where they are used in chemical reactions or used to synthesize new molecules in the body to allow tissues to grow and repair. Task: what are the different food groups? Extension: what is each food group for in the body? The main food groups are carbohydrates, proteins, lipids, which are fats and oils, and vitamins and minerals. Carbohydrates are for energy, as they provide glucose, which can be used in the process of respiration in the cells to release energy. Proteins are important for growth and repair of the body, as well as normal functioning of the other body systems. This is because they provide amino acids, which are used by the body to build new proteins. Proteins in the body can be structural or functional. Structural proteins make up our body's structure and form, such as collagen, the protein in bones. Functional proteins facilitate chemical processes in the body, such as hormones and enzymes. Lipids provide fatty acids and glycerol, which is used by the body to build molecules and also provide energy and insulation. Task: what are the organs of the digestive system? The organs of the digestive system are the stomach, liver, gallbladder, pancreas, and intestines, although the food does not pass through all of the organs. Digestion begins in the mouth, where the food is chewed, which increases the surface area and breaks it up, ready for swallowing. The food in the mouth is also mixed with saliva, which moistens the food to make it easier to swallow. Saliva also contains some enzymes. These enzymes are called amylase and are a type of carbohydrase enzyme. This means that they break down carbohydrates. From the mouth, the food moves down the esophagus and into the stomach. The stomach is a large organ that secretes hydrochloric acid and an enzyme called pepsin. Pepsin is a protease molecule that breaks down protein. Pepsin works best in acidic conditions, so the acid helps to provide the optimum conditions for protein digestion. The acid also kills any pathogens that may be in the food. The muscular walls of the stomach contract to churn the contents, mixing the food with the pepsin and hydrochloric acid. After the stomach, the food enters the small intestine. Here, all different types of enzyme are mixed with the food, along with bile from the gallbladder. The bile is alkaline and helps to neutralize the stomach acid as well as emulsifying the lipids present. The enzymes in the small intestine are carbohydrase, protease, and lipase enzymes. This means that all of the food types can be digested. The enzymes are made in the small intestine and in the pancreas, from where they enter the small intestine through a duct. Any molecules of food that are small enough will pass through the walls and into the bloodstream. The food that is left enters the large intestine, where any excess water is absorbed. Any solid food particles left are egested from the body through the anus as feces or poo. Task. Draw and label a picture of the digestive system to show the stages of digestion. Extension. Which organs of the digestive system does the food not travel through? Although part of the digestive system, the food does not travel through the liver, gallbladder, or pancreas. These organs, however, do have important functions. The liver produces bile, which helps with the digestion of lipids. The fats and oils are emulsified, which means their surface area increases. The bile is stored in the gallbladder and then enters the small intestine through the bile duct. The pancreas is important in enzyme production. Carbohydrase, protease and lipase enzymes are made by the pancreas. The carbohydrase enzymes produced by the pancreas are called pancreatic amylase. It's different to salivary amylase as it breaks down large sugar molecules into glucose, which can be absorbed into the bloodstream. Enzymes are biological catalysts that speed up the process of digestion. Enzymes are a specific shape which fits a specific molecule. For example, carbohydrase enzymes, such as amylase, fit a carbohydrate molecule, such as starch, to break it down into sugars. The molecule that the enzyme fits is called the substrate. As it combines with the enzyme, it is called the enzyme-substrate complex. The active site of the enzyme is where the reaction takes place, and once complete, the products of the reaction disengage with the enzyme and move away. This is called the lock and key hypothesis, as the enzyme and substrate fit together like a key into a lock. Task. Fill in the spaces in the following sentences. Digestion is the process of breaking down large food molecules into smaller molecules that are soluble. Enzymes help to break down the food because they catalyze or speed up chemical reactions. Extension. Where in the digestive system are each type of enzyme produced? Carbohydrase enzymes that break down carbohydrates into glucose are made by the salivary glands and the pancreas. They act on the food in the mouth and the small intestine. Protease enzymes that break down proteins into amino acids are produced by the stomach and the pancreas, and act on the food in the stomach and small intestine. Lipase enzymes are produced by the pancreas. They break down lipids into fatty acids and glycerol and act on the food in the small intestine. There are some factors that can affect the activity of enzymes. These are pH, temperature, and concentration of enzymes. All enzymes have a pH that is the optimum for that specific enzyme. When the pH is not at the optimum, the shape of the enzymes changes and it will no longer fit the substrate. As temperature increases, enzyme activity increases, as the particles have more energy. However, when the temperature gets too high, the enzymes are denatured. This means they can change shape and no longer fit the substrate. When an enzyme is denatured and changes shape, it no longer works, and the reaction will slow down to a minimum. When there is a high concentration of enzymes, there are lots of active sites available for the substrate to fit with, so the breakdown of a food type will occur quickly. When the enzyme concentration is lower, then the rate of the reaction will be lower.